because it's one of the last two for this academic year. So today we have Kinga Bujakowska, and next week we will have Cher and G from here. So welcome. And as usual, if you have any questions, comments, please let me know and be ready to speak when prompted by the moderator. Moderator for today is Chris Palczewski. And with that, let's introduce our speaker.
Hi, good morning, everyone. I'm Rui Luo, a graduate student from Dr. Ray Chen's lab. It's my honor to introduce our distinguished speaker, Dr. Kinga Bujakowska, an assistant professor of ophthalmology at Harvard Medical School and a principal investigator at Massachusetts Eye and Ear. She's a leading investigator in the field of IRD genetics and functional genomics. Dr. Kinga Bujakowska earned her PhD in molecular genetics from the University College London, where she studied the disease mechanism of a specific form of retinitis pigmentosa caused by mutations in the splicing factor gene PRPF31. She did her first postdoctoral training in the Institut de la Vision, France, where she was involved in mapping new genetic defects in patients with retinitis pigmentosa and congenital stationary night blindness. She later completed her secondary postdoctoral fellowship at Massachusetts Eye and Ear before joining the faculty. Over the course of her career, Dr. Bujakowska has contributed to the discovery and characterization of multiple disease-associated genes. Her research has significantly advanced our understanding of the contribution of non-coding and splicing variants to IRD pathogenesis. Her group has developed high-throughput functional splicing assays and complementary genomic analysis frameworks that enable systematic interpretation of variants of uncertain significance. Her laboratory also developed an in vivo retinal CRISPR knockout screens and created the Mendelian analysis toolkit. She also contributes her expertise as a member of the ClinGen expert panel of IRD gene and variants curation. Now, please join me in welcoming our speaker, Dr. Kinga Bujakowska.
Thank you so much for this very kind introduction, and thank you for your invitation to participate in your seminar series. It's a great honor and pleasure to present. So I'm going to talk about what I've been doing in the last many years, and so it's searching for elusive causality in retinal disease. So I've been focusing on inherited retinal degenerations, and I'm sure you are all familiar with those diseases, but those IRDs encompass quite a few different phenotypes: rod cone degenerations or RPs, macular degenerations or early onset retinal degenerations, commonly called LCA, and they can be syndromic or non-syndromic, and the prevalence varies and is between 1 in 2 to 4,000 individuals worldwide. So I'm going to discuss today 3 themes. I'm going to talk about new disease gene associations, about the contribution of structural variants in IRDs, and variants affecting pre-mRNA splicing. But to start, I wanted to point out that IRDs or retinal diseases are extremely genetically heterogeneous. In the recent publication from the Revolta group, they curated 470 genes that will lead to various retinal diseases. They include IRDs, syndromic and non-syndromic, as well as optic atrophy genes. And what is kind of striking is how much, the genetic heterogeneity, but also how much overlap there is between the different phenotypes and the syndromic and non-syndromic forms. And you may not see it if you're sitting far away, but all the genes that are underlined, they cause syndromic and non-syndromic diseases. And the more we learn about IRDs, the more overlap there is. So it makes sense that instead of calling those diseases by those archaic old names like LCA2, for example, it would be better to call the disease by the gene and the mechanism. So, for example, RP65 related to recessive retinopathy, or CEP290 related to ciliopathy, rather than LCA2 or LCA10. And if you're interested, I really invite you to look at ClinGen. ClinGen has done important work in classifying those disease entities and gene disease validities. And as you can see, the work is ongoing because there's so many genes associated with these diseases. So you may think, okay, we know so many genes now, surely we can genetically solve all of the IRD cases, but it's actually not the case. So when we look at, this is a sample of 3,000 individuals from Masai Near. And after performing genetic testing, we can give confident genetic diagnosis to about 56% of the cases. And obviously, this will vary depending on the cohort you are studying, but this is kind of our cohort. And the remaining cases, they either have variants of unknown significant or no genetic leads. So, and you see in this pie chart, those are the prevalence of different genes. You know, the most prevalent genes are the very well-known culprits. And then the more genes we discover, the rarer they become. So what could be the reasons for the misgenetic diagnosis? Obviously, one of the reasons is there are still more genes to be identified. Oh, I'm sorry. Okay, there. And then, so my first story is will be about expanding the IRD gene landscape. Do you guys have something change in my? I think I'll just continue. Anyway, so here I'm going to tell you about a new disease gene discovery where biallelic variants in PRKCZ lead to late onset rod cone dystrophy. And we have gathered through collaborative study 10 families in which we identify biallelic variants in this gene. Most of those variants are loss of function variants, with one exception, missense variants, and they lead to late onset retinal disease. What is PRKCZ? PRKCZ gene codes for protein kinase C zeta, and this is part of the families of protein kinase C, and those are central regulators for cellular signal transduction, and they're basically involved in every aspect of the cell life and involved in every aspect of human health and disease. There are three classes or three types of PKCs: classical, novel, and atypical. And our PKC zeta belongs to the atypical class of PKCs. And what differs them is the different regulatory domains. So the classical PKCs are activated by calcium binding, diacylglycerol, and phospholipids. Novel PKCs, diacylglycerol and phospholipids, and the atypical PKCs are activated by phospholipids and proteins with the same PB1 domain. And they all contain kinase domains. So, and more specifically, PRKCZ or PRKC zeta has been implicated. There's not much known about the function of this gene, but so far has been implicated in cell polarity, mitochondrial signaling, neuronal survival, and epithelial organization. This gene is expressed in the retina as well as in other tissues, and notably, it's quite highly expressed in the brain, lung, and the testes. Yet the patients with mutations in this gene have only non-syndromic late onset IRD. So we were curious to understand, like, why would this widely expressed gene with biallelic loss of function variants cause this very specific retinal phenotype? So we looked a little bit more in more detail at the different transcripts, and there are 4 major transcripts that this gene has, and then where our mutations are positioned. Most of them are clustered in the 5' part of the gene, with one exception being more in the middle of the gene. And you will notice that most of them will affect the longest transcript, which codes for the long protein isoform that contains PB1 domain that is activated by the interaction with other proteins that have this domain. C1 domain is activated by the phospholipid domain, and then this is the kinase domain. And when you look at compare the peripheral retina and the brain and the contribution of those four different isoforms, you will notice that the two longest isoforms are more important in the retina, actually, than in the brain. And this could be the reason why the phenotype is so specific in the retina. If you look at the single-cell RNA sequencing from the retina, you will notice that it's particularly highly expressed in the rods, which also corroborates the fact that this is a rod cone degeneration. So we wanted to understand a little bit more like what is affected on the cellular level, and we studied patient fibroblasts, and this has been done in the lab of Marco Tartaglia in Italy. And so when he compared the ciliogenesis in control and patient fibroblasts, he observed stunted cilia, or in the patient fibroblast lines. So they have less cells that have the full-length cilia and more cells that have stunted or absent cilia. Since this gene has also been implicated in mitochondrial function, we looked at the mitochondria in the control and patient fibroblasts, and we have observed increased fusion of the mitochondria that is kind of seen here by this mitotracker, more elongated mitochondria compared to the control. And this could be a compensatory stress response. So I hope that you will agree with me that this is a very likely new IRD disease gene, and currently our manuscript is in review. So in recent years, we were grateful to contribute to a few new disease gene discoveries, and altogether, these genes, they contribute to, in our cohort, to this increasing solving rate of 1.6%. So we are still very far away from completing our misdiagnosis pie chart. So what could be other possibilities? So other possibilities is that we are missing some variants, some elusive mutations in already known IRD genes. And for example, a big class of such variants could be structural variants. So what are those structural variants? Those are variants that are larger in size, classically more than 50 base pair in length. And those could be deletions, duplications, insertions, as always, balance structural variants of translocations, so from to a different chromosome, for example, inversions, or kind of more other SVs, like complex SVs that can contain any combination of those previous categories, or repeat expansions. And ideally, you would have long-read sequence data that would be, kind of easy to resolve those. But since it's still quite expensive, we rely on short-read SV calling. And I'm going to explain a little bit how it works so that you can understand my slides later on. So with the paired and short-read whole genome sequencing, we rely on the fact that during the library preparation of the DNA, of patient DNA, we fragment the DNA into this known between 300 and 500 base pair fragments, and we sequence those fragments from both ends. So here are the example reads. So those are the fragments, and we sequence 150 base pairs on both ends. And since those reads are tagged, they are paired, when we map them to the genome, we expect them to be to both ends to be at the expected distance that would correspond to the size of the library. So if in the mapping the distance is expected, we predict there's no structural variation. If there's a deletion, the reads will seem to be further apart than there should be. And for example, if there's an inversion, those reads will point to the same direction. So what is also important, that the paired and they point in the opposite directions, and during inversion, they will point in the same direction. So how do we leverage the data to resolve a choroideremia family? So this is a 3-generation family that with choroideremia in the grandfather and 2 half-brothers. They have undergone numerous genetic testing, and it was always inconclusive. So, and this, well, we performed whole genome sequencing on this family, and the algorithms are not always accurate. So sometimes you need an astute eye of a researcher. And here, this is work that was performed by Jinu Han, a collaborator from South Korea. And he observed that there are those actually reads, if you can see, I'm not sure if you can see, but those are the teal reads. Those are paired reads that point in the same direction, which would indicate an inversion. And the blue reads point also those are pairs that point in the same direction by the opposite direction. So we have two indications of an inversion and a deletion. So basically, what's happening in this particular case, there is this previously unseen inversion that breaks the CHM gene in intron 12. That's why it hasn't been seen or observed before, and an upstream with the upstream sequence. So altogether, there is about almost 6 megabase inversion with 36 kilobase deletion. And this is the likely cause of disease in this patient. So we have seen a lot more of such cases, and altogether, we estimate that between 5 and 10% of our cases are solvable with whole genome sequencing short read structural variation algorithms. And obviously, there may be more hiding that we are not seeing because we need long read technologies. So what are the other possibilities? So other possibilities that we may be missing are some regulatory variants that we don't know how to interpret, and also intronic and exonic variants of uncertain significance that we currently do not know how to interpret. And those VUSs is a common burden in clinical genetics, not only in IRDs, but in all other Mendelian diseases. And here is just a snapshot from ClinVar. ClinVar is a repository of rare variants seen in patients with various Mendelian diseases. And you can see that half of all the variants deposited in ClinVar are of uncertain significance. So there's a lot of ways that you could resolve those VUSs, and in the OGI, such efforts are undertaken. So the group of Lizzie Rosen is looking at the structure-based network analysis. So basically, she's interested in how missense variants change the structure of the protein and hence the function. And also, Dr. Jason Commander, he studies how missense variants affect function of the protein, for example, trafficking of rhodopsin. And in my lab, what we asked ourselves is, can a substantial number of those VUSs affect pre-mRNA splicing? And these could be events that lead to exon skipping, altered 3' and 5' splice sites, or cryptic exon inclusion in the intron. And many variants are known to alter core splicing signals, and some of those are already kind of well characterized. Those are variants that are close to the 5' donor side and 3' acceptor side, but we wondered, can we study those variants that are further away, that are non-canonical splice site variants? We can investigate those effects in patient cells. And for example, if the gene is expressed or expressed well enough in for example, blood or fibroblasts. And here's an example of a study that I've been doing in collaboration with Bruno Popowski and Garvin Arno. We have identified variants in PRPF31. This is an essential splice site variant, so it's our control, and a -12 variant that was of unknown significance. We have isolated RNA from peripheral blood cells and performed long-read sequencing. And here what I'm presenting is, those are called sashimi plots. And for people who are not familiar, here at the bottom, you have the structure of the gene with the exons of PRPF31. And those bars indicate reads, the map to the exons, and those jumping hoops are exon junctions. And the number indicates the number of reads that pass from the junction from one exon to another. So in the control, we see a perfect match to the known gene. In our -2 control, we see that there is an alteration of one of the splice sites. This is where the mutation is. And when we zoom in, we can see that the acceptor side has been changed, and it leads to the extension and inclusion of part of the intron in the exon, and this leads to a frame shift. And then in our case, in the -12 C2A change, we see that there is an extra exon here. And then when we zoom in, there is inclusion of a cryptic exon that actually has been predicted in some transcripts, and also an extension, similarly to here, extension of the splice site. So altogether, that leads to a frame shift and degradation of that allele. So both mutations are likely disease-causing in those patients. So what do we do when we don't have either the blood is not accessible to us, or the gene is not expressed in the blood? So we can perform a high throughput splicing assay, and this is similar to what the group of Ray Chen in your institution has been also developing. So basically, the assay relies on a split GFP, into which we clone a pool of sequences that have exons or cryptic exons, putative cryptic exons, and then M-chair is our reference fluorescence. So if the sequence is recognized as an exon and is included, then it will lead to a disruption of GFP. And if the sequence is not recognized as an exon, it's skipped, and that leads to a reconstitution of GFP and the fluorescence. So in order to take advantage of this green, no green readout, we should really have only one construct per cell. And to do that, we use a landing pad cell line. Those are HEC 293 cells with the landing pad at the safe harbor AVS1 locus. The locus contains ATTP recombination site and a geneless promoter. Our construct contains ATTP and the promoterless puromycin. And when we co-transfect these plasmids with the BXB1 recombinase, the correctly integrated construct will lead to the promoter will drive the puromycin resistance, so we can actually apply the puromycin gene to select our cells. And then after multiple passages, when we do 10 passages to be sure that there is no episomal plasmid DNA, so that the fluorescence comes only from the integrated construct, we perform FACS sorting. And here's also from one of our experiments, we can see two populations of GFP negative and GFP positive cells. GFP negative indicate exon inclusion, and GFP positive indicate exon skipping. And we perform sorting of these cells, genomic DNA extraction, PCR, and applicant sequencing, and then we count the exact reads that are mapping to each of the oligos in both of the pools, and those are normalized by the sequence depth and the sort fraction, and those normalized oligo counts can be fed into the equation that will calculate the percent spliced in. So those are normalized GFP negative counts over GFP positive and GFP negative, which the PSI in effect is a fraction of mini-gene transcripts with test exons included. So first we wanted to see, is the method working? So if we look at all the reference exonic sequences and reference intronic sequences. So in the reference exonic sequences, the inclusion should be very high. So, and this is what we mostly observe, the PSI percent spliced in is high. And for the intronic reference sequences, we assume the PSI should be very low, and this is exactly the case. So most of those intronic sequences are not recognized as an exon. There are some sequences for which, as you see, it's not working, but most of the references work as expected. So then when we looked at the controls, we see that roughly half of our essential splice site controls, so those are acceptor and donor site changes, will lead to the skipping of an exon, but there's quite a lot of them that show no effect. So basically what is happening, like if a donor or acceptor leads to aberrant a shift in the splice site, we would not be able to distinguish it from the references. So in order to resolve this problem, we are performing a secondary experiment in which that involves a transient transfection, and we test what is included actually between the two GFP exons, and we compare it to the reference, to the gen code. So we can see, we can distinguish between the reference exon inclusion and the altered exon inclusion. And for that, we kind of introduced a new term called percent reference exon splicing. So if there is high percent reference exon splicing, as we would expect for the references, you have PRES of 1. And if the included sequence is aberrant, you will have PRES of 0. So you could see that most of those essential splice sites will have a very aberrant inclusion of the exon. So it's either shorter or longer. So if you to be able to kind of take advantage of both assays, we integrated the PSI and the PRES scores into one aberrant splicing score, AABS. So now with this score, we are able to capture all of these events. And again, the aberrant splicing score for the reference exonic and intronic is low because most of them splice as expected, with some exceptions. And most of our controls, of essential splicer controls, have high aberrant splicing score because they either lead to skipping of an exon or aberrant sequence inclusion. So now we were ready to perform our experiment. So we had variants from IRD cohorts from Massainier and our collaborators, as well as variants nominated by the Rare Disease Network, GREGOR, that stands for Genomic Research to Elucidate the Genetics of Rare Disease Consortium. In our libraries, we had about 1,000 essential splice site controls and almost 4,000 test variants. And to be able to really interpret the results, we always compare it to our references. So we'll be looking at delta aberrant splicing score, which is absolute value of the aberrant score of the variant minus the reference. And with this, I'm just going to show you that the distribution, if you look at the green, this is the distribution of our test variants, and in red is the distribution of our essential splice site controls, and you see, as you saw before, most of them have a high aberrant splicing score, and for our variants, we took a cutoff of delta ABS of 0.5, which to us seems biologically informative that at least 50% of the transcript will show some aberrant splicing. And in this category, there's about 6% of the test variants showed aberrant splicing, a significant aberrant splicing score. So next we wanted to compare, to understand how does our assay compare to the AI prediction algorithm, and then the most commonly used algorithm in the clinical genetic testing is a Splice AI. Splice AI has been trained on the pre-mRNA sequences, on the gen code and gene structure, and they have been tested as well on the other part of the genome, on the gene structure and the GTEx data. So basically, with the Splice AI, if you have a rare variant, it will predict whether that variant leads to a splicing defect. So in this case, it creates a variant that is not far from the exon, creates a alternative acceptor site, so acceptable gain with the delta score of 0.9, so 994, so quite high, and that it creates a loss of the original acceptor site. So basically, we apply this to our data quite commonly. And but there are those different scores that you can apply, and then different labs have different thresholds. And for the purpose of my comparison, I choose the delta score of 0.5. So when I looked at my data, so if you plot the variants that I studied, and on the y-axis you have splice AI max, maximum score given, and on the x-axis you have delta ABS, so aberrant splicing score, you'll see that 85% agree of the variants. I mean, the results agree. 73% have splicing effect below our threshold and 12% above, and 15% disagree. But when you look at kind of more closely at the SPICE AI predictions, they predicted that sensitivity of 60%, a little bit over 60%, and validation rate of 60%. And this is basically what we see here. Of all of the variants that we validated that cause a significant splicing defect, only 60% were predicted by SPICE AI. So that's our sensitivity. And of all the variants that were predicted to at this threshold to cause missplicing by SPICE AI, 66% validated with my assay, which is also very close to their prediction, their original prediction. So it's kind of interesting how this very artificial small mini gene splicing assay leads to actually corresponds precisely to that to those predictions from the original paper. Another way of looking at the data is, well, what we could have missed. So again, depending on the thresholds that you set in your laboratory, if you only look at variants that have any score of SPICE AI, you could have missed 37 of the variants that actually lead to splicing defects and potential diagnosis. And then if your threshold is 0.2, you would miss 84. And those are the different thresholds. So the variants that we tested that show the significant effect on splicing, most of them were intronic, but there were some missense and synonymous variants as well. So just wanted to give you two examples of the genetic diagnosis confirmations using that assay. And one of them is a patient that has a retinopathy and also has inconclusive genetic diagnosis. The patient had two variants, one a missense variant and a synonymous variant in FLVCR1 gene. Both of variants are classified as VUSs. The missense variant VUS leaning towards likely pathogenic, and the synonymous variants was a VUS leaning more towards benign. And all the algorithms showed that it's likely benign. However, when we tested that variant in the high throughput splicing assay, we saw a very significant effect on skipping. So here, 95% of those mini gene transcripts showed skipping. And when we validated it in the large genomic context, we have seen the same. So this is a reference, this is a variant, and we can observe skipping of that exon. The exon is out of frame, so this allele, and the variant is actually very close to the edge of the exon. So this allele basically is a loss of functional allele. We did not see any effect on splicing of the missense variant, but we still think that this case is likely solved by those two variants. FLVCR1 is a kind of is quite an interesting gene because it can lead to a broad spectrum of phenotype. So it's a choline and ethanolamine transporter. It can lead to isolated retinal disorder, as in the case I presented to you, but also to ataxia and sensory neuropathies. And those two are mainly when there's a missense and a loss of function variant. And more recently, we contributed to a manuscript that shows that two loss of function variants leads to severe developmental disorders. And here I just wanted to show you one case where a patient with a severe phenotype had one clearly loss of function allele, and another allele was a rare missense variant. When they tested the variant in terms of its ability to transport choline and ethanolamine, they haven't seen any differences between the reference, the wild type and the variant. So that's when Daniel Calamay asked me if I could test the variant in the splicing. And we've seen a high effect on exon skipping with the aberrant splicing score of 0.5. It also had an elevated splice AI score. And this we confirmed in larger genomic context, where here you have a reference and you do see a little bit of skipping, but the skipping is exacerbated by the missense variant. So it's actually important to point out that a combination of those functional assays is often necessary to prove the pathogenicity of variants, and we shouldn't rely on one assay, for example, to demonstrate or to draw conclusions that a particular variant could be benign. So here I would like to conclude what are the key findings of our studies. So novel IAD genes continue to emerge, and then there will still be likely many more genes to be discovered. Structural variants do account for a substantial number of misdiagnoses, and also probably that's an underestimation. I have not shown you regulatory variants, but we have also have contributed to some discovery of some of those. And then those splicing VUSs also have a significant contribution, and those are sometimes they would be they're missed by the prediction algorithm. So it's worth testing variants that have no predictions. So I do feel like we are in the area where the functional genomics is redefining the molecular diagnosis, and those remaining unsolved cases will need a deeper interrogation of the genome function, for example, understanding the non-coding regulatory elements and also genome structure. I'm sure there's a lot of variation hiding there. I mean, a lot of causal variation hiding there. Also, it's important for us to, as a community, to work on the improved structural variant detection, and I think this will involve long-read sequencing technologies. High-throughput functional assays, I think, will continue to be very instrumental in defining pathogenicity of many variants. We all implement different deep learning prediction algorithms, and then as those high-throughput functional assays improve, I think those prediction algorithms will improve as well. And I think it's very important to work collaboratively to resolve those very rare genetic causalities. And this brings me to my last slide, where I would like to acknowledge my coworkers, my wonderful lab members who keep the work in the lab going. I work in the very nourishing, great environment at the Ocular Genomics Institute, and I would like to thank all of the collaborators with whom we have the common goal is to be able to provide them diagnosis for all those cases, so that one day therapies can be developed. I would like to thank also all my funding institutions, and I was very fortunate to have been supported by a lot of grants. Thank you.
Thank you, Kinga. I have two questions. One related to PKC-zeta. So obviously the PKC will not have effect, except of its activity will have effect on degeneration. So what is the potential substrate for phosphorylation? Did you guys consider to explore who is the recipient of the phosphorylation from PK-zeta?
That's so no, not yet. So this is I think that would require some work with retinal organoids. So we have collected some IPS lines for the patients, and we are hoping to study that. But no, we haven't looked in detail.
The second question I have before I pass it to my colleagues here. There was a lot of, I really ask usually Ray, and he's annoyed by my questions about that. How about the multigenic, like the Draja discovered at least 2 genes involved in disease. So are they multigenic, and those are very difficult to diagnose?
Yes, so those more digenic or polygenic, they're kind of difficult to diagnose. There's really like a handful of publications because you would have to the families that we work are very small, and to prove that this is truly due to this digenic inheritance, it's just very hard based on just the family kind of Mendelian genetics. There was like a paper in Katsanis with the BBS genes and the Dryja, but somehow there were not so many cases after that. I think now, as we are more working on sequencing of the larger cohorts, like just populations, and then looking at the deeper phenotyping, we are discovering that there are people in the population that carry genotypes that are associated with sometimes quite early onset retinal diseases, and yet they seem to be unaffected. And I think as more of those kind of people are discovered, and those maybe we'll be able to discover more what are the modifiers. I'm thinking about it more as a modifiers of disease rather than kind of digenic inheritance. So very likely there are other factors, genetic factors, that influence the expression of the disease, and we are considering that, but it's still I don't have like a very concrete answer to you to tell you what those are. This is like a new frontier of IRD genetics.
Thank you so much for your wonderful talk. I'm Gulab Zode from UCR1. I have a question about the PKC and the mito tracker you showed. Using the mito tracker, it's kind of really hard to say whether it's the fusion that is affected. And the way I see it is that in fact the PKC fibroblasts, they look like elongated mitochondria, which can be actually good because there are more specific markers for the fusion, DRP1 and others that you can conclusively say. Also, if the mitochondria is affected, wouldn't most of the other cells would be also affected, like ganglion cells, because they really require mitochondria and transport and all that. So thank you.
Yeah, that's a great question, a great suggestion for future studies. So we just were looking for, can we see any phenotypes in the fibroblasts, and that's what we've observed, that the mitochondria are elongated, and that's kind of my hypothesis that the balance between fission and fusion is more towards fusion, which could be kind of protective, but at the same time, patients, they do not have other phenotypes. Or maybe we would have to really very deeply characterize them clinically to observe other phenotypes. But there was nothing striking. Some of the patients had hearing anomalies as well, but we were not sure whether it's related to that gene. So I feel like this is because this is a new genetic discovery, and that there's a lot more obviously to be understood about this gene and its role in the retina. So I think those are all great suggestions for future studies.
Okay, let's go to the Zoom now. Mariana Wiener, do you have a question, comment?
I posted my question. So Kinga, amazing work. Thank you so much. Have you checked alpha genome, or do you have a plan to do that for alpha genome? Because my results correspond a lot with
Alpha
discrepancies.
Mm-hmm. Yeah, no, and I think that's definitely we should do that. But we haven't kind of compared systematically at least.
Mm-hmm. Thanks.
Thank you very much for your time and insightful work. So I have a question regarding the VUS. How often should a patient with a VUS undergo like reinterpretation of their data or undergo like whole genome seq? Also, who should be responsible for the choice of analysis? As a clinician, we just send our sample to the company, and then we just rely on their pipeline or their analysis.
Yeah, so I guess with those diagnostic genetic diagnostic companies, I haven't been working closely with them, but they obviously they will have some kind of standard pipeline, and I would suspect that they do reanalyze periodically data. I think in general it's good to reanalyze data periodically because there's obviously new IRD genes, there's new interpretation, there's new ClinVar, there's more and more updates in ClinVar, and then we can also rely in the providing the genetic diagnosis, we rely on the fact, has this variant been seen before in the variant scoring. So all those things kind of keep improving, keep moving. But so yeah, if you had your own data, I would say revisiting the data every 6 months, if it's not too burdensome, would be a good idea. And the clinical labs, I guess they do that anyway, but maybe they should be prompted by clinicians to do so. Some labs, maybe they could provide actually the raw data for you to analyze it, in collaboration with the genetics center. That's another possibility.
I think that's great. Very nice talk. So I have a lot of questions, but I just picked two simple ones. So the first one is just clarification on the high throughput, splicing assay you have. Are those candidates are near exons, or there's some of them are deep intronic? And then if you do have both, what's their behavior looks like in terms of relative to a SPICE AI score or relative to the PSI change? Do they show difference? And the second question is, I noticed the slide you show the PRPF, splicing, fibroblast splicing, the rare splicing about 20 to 30% of the normal ones. You still have the normal splicing, and you think they're pathogenic. So how do you determine, like, if it's 20% missplicing, how bad is it?
Okay. So I'll ask the last question first. I'll answer. So for the PRPF31, that was in the actual blood, but it doesn't matter whether fibroblasts or blood. So the allele, most likely that allele is being degraded by NMD, and so then you don't expect to be half and half, right? If you expect that variant leads to frame shift and the allele is degraded. Sometimes you can actually see that if there is a SNP, if there's a synonymous or a coding SNP, you can see the allelic skew. In these particular patients, actually, there were no heterozygous SNPs to actually prove that. But the assumption is basically that the allele is degraded. For the first question about, I did have in my libraries deep intronic and exonic variants. How did they, and it's true that in my analysis, I lumped them together in terms of the SPLICE AI scores. In general, the SPLICE AI scores are lower for the intronic or deep intronic variants, but I don't have like a good breakdown of those variants that I studied. Where do they? But I think it's a good idea to kind of separate those and demonstrate. But in general, the confidence in SPLICE AI, I feel, is lower in the deeper intronic variants than in this kind of periexonic variants.
Thank you.
Hello?
Yeah.
Is it working? Okay, cool. Hi, wonderful talk. I'm not really in the field of gene exploration, but from knowing that there's so little cases of these rare diseases, I guess to me it would make sense to look into mouse models instead of organoids. And I was wondering why you wouldn't explore choroid-specific late onset disease on a mouse like LORD mice or other models and just see how it progresses. Just curiosity.
No. So we do model IRDs in the mouse, so very often, but sometimes a mouse does not show a phenotype or shows a very late phenotype. So for example, the PRPF31 gene that I showed that showed this in missplicing, the phenotype is very, very late onset and very you have to wait 2 years. So that's where organoids, more kind of human model is important. For the PRKCZ gene, very possibly you're right that here studying in the mouse could answer some of the questions, like what are the targets, and what are the interactions, and why is this gene so important in the retina. So that would be a good idea.
Hi, Kinga. This is Rafał from Chris Palczewski Lab. Nice to meet you
Hi.
again. I have a question. I'm not a clinician. I work in the lab, but I try to imagine once you have your diagnosis and proposed mechanism of the disease, what do you offer to patients? Can you tell them we're working on the gene therapy or genome editing? What can you tell the patient, like moving forward? What's the plan? Thank you.
So actually, I'm not a clinician myself, so I don't have that burden to tell the patients. But I imagine, talking to my colleagues, depending on the gene, they explain that there's many efforts, for various genes that are being done to develop gene therapies. And so the first thing is always having a confirmed diagnosis, and that's a very important first step. And then if you happen to be in the gene that there's anything in the works, obviously that's good news. But many, as you saw from my pie chart, many patients have extremely rare variants in very rare genes, and at the moment, I guess things like therapies like protecting degeneration of gene agnostic therapies, that would be the best option for them, protecting the cones, for example, if it's a rod cone degeneration.
Dr. Brown, what do you say to your patients?
Yeah, hi Kinga, good to see you.
Yeah.
May remember me, I'm Rob's friend. Yeah, so with these cases, it is frustrating because there's not a lot of intervention that we can offer, but primarily, all these patients are looking for hope, so primarily focus on not losing hope. And the thing that I always tell them is, even in the 15 years that I've been at this, which is not that long in the spectrum of IRDs, you just say, this is the time of major changes, and I'm really excited for what is in the pipeline in the coming years.
Wonderful. Thank you for.
Thank you, Andrew. So I want to just to maybe close today's seminar with another thought that we're all aging with different rate, and clearly you will have cases where even members of the same family with the same mutation will age at different rate, and so the diagnosis will be quite difficult because of progressive nature of many of those retina diseases, and how we can overcome that problem that one person, it goes back to the example that I remember from University of Washington, and I was saying that story about 10 times already, so don't be burdened with that. But there were two sisters, and they both had a P23H mutation, and one was driving the other to see a doctor. So one was almost unaffected, very, very minor changes, where the other progressed very quickly. And that will really impact the interpretation of genetic tests, wouldn't it?
Yes, that's why there's a big debate. Should we test people pre-symptomatically? What does it mean to test, kids, right? Or before they have symptoms, because maybe they'll never develop the disease. For the rhodopsin P23H, yes, it's a very well-known fact that you have a huge variability in phenotypes, and this is one of my interests is to study modifiers of this genetic modifiers of the gene of the severity of those, and we have been developing some tools to use CRISPR in the mouse model with P23H, which actually the mouse that you have generated in your lab, Chris, and trying to figure out knocking out or which of the genes can actually ameliorate the phenotype. So I think there's a lot of done for like particular genes. Those mechanisms could be gene specific, for example, something, that helps to mitigate the deleteriousness of the P23H, relapsing mutation. But there could be also some general factors that actually enhance survival of the cells. So I think both probably would be at play, and it would be very interesting to study those.
All right. Thank you, Kinga. Let's please join me to thank our speaker.
Thank you. Thank you for having me and for listening. And yeah, I look forward to seeing you at the conferences.